Welcome to 2-Minute Anesthesia in Critical Care. Let's review antibiotic resistance mechanisms. Antibiotic resistance is defined as the ability of a bacteria to evade the effects of antibiotic treatment, which can occur through different mechanisms. Why is this important? Antimicrobial resistance is a central global health issue. Estimated that bacterial resistance was responsible for 1.3 million deaths in 2019 and contributed to 5 million deaths. The misuse and overuse of antimicrobial treatments in humans, animals, and plants are the main driver in the development of drug-resistant pathogens in all countries. Penicillin was discovered by Fleming in 1928, but it wasn't until 1940, several years before the widespread introduction of penicillin, a bacterial penicillinase or beta-lactamase was identified. Following widespread use, resistant strains became apparent, resulting in the need to change the chemical structure of the penicillin to prolong the degree of effectiveness. Broadly speaking, antibiotics have four main mechanisms of action. These include inhibition of multiple pathways involved in cell wall production, protein synthesis, metabolic pathways, and nucleic acid synthesis pathways. In addition to other mechanisms, resistance can affect all of these pathways. For example, ribosomal modification can prevent the interaction with key antibiotics. These include aminoglycosides and macrolides. There are two main types of resistance, natural and acquired. Natural resistance can be further divided into intrinsic, i.e. always expressed, or induced, becomes effective once exposed to an antibiotic. The most common mechanism involved in intrinsic resistance are reduced permeability of the outer membrane, especially lipopolysaccharides in gram-negative bacteria, but also the presence of key efflux pumps which export antibiotics from inside to outside the cell, thus preventing the onset of action. Acquired resistance refers to the acquisition of genetic material that confers resistance. This can be achieved via multiple routes involving transformation, transposition, and conjugation. This may lead to transient or permanent antibiotic resistance. Plasmid-mediated transmission is the most common method, leading to a range of effects involving target modification, inactivation, and efflux of antibiotics. Antimicrobial resistance pathways lead to four main groups: limiting the uptake of the drug into the cell, modifying the drug target, inactivating the drug, or active drug efflux. We will now discuss each of the pathways in a little more detail. First of all, limit or prevent uptake. There's a range of natural defense mechanisms which limit antibiotic drug uptake. For example, gram-negative bacteria have two membranes compared to gram-positive, which just have one. While some bacteria, including mycoplasma, lack a cell wall, preventing all drugs which act on a cell wall becoming effective, i.e. beta-lactams and glycopeptides. In bacteria with a large outer membrane, substances can still enter via porin channels. These porin channels can result in modification in structure or number, thus inducing resistance. And finally, biofilms may form. Secondly, modification of targets. For beta-lactam antibiotics, alteration in the structure or number of penicillin binding proteins can induce resistance. Methylation of ribosomes can induce resistance through the inability of the drug to bind. Mutations have also been observed in a range of enzymes, including gyrolase, topoisomerase, and dihydrofolate reductase, which decrease or eliminate the ability of the drug to bind to these components. There are two ways in which bacteria can become ineffective: by actual degradation of the drug or transfer of a chemical group to the drug. And finally, there's a range of drug efflux systems which remove the drug from the cell. These include the ABC cassette, MATE, and MFS. Beta-lactamases, also known as penicillinases or cephalosporinases, inactivate beta-lactam drugs by hydrolyzing a specific site in the beta-lactam ring. This causes the ring to open and therefore is unable to bind to the active site. Beta-lactam enzymes are classified based on their structure and functional characteristics. Commonly used beta-lactam inhibitors include clavulanic acid and tazobactam. Examples of drug resistance in ICU include methicillin-resistant Staphylococcus aureus, or MRSA, Extended spectrum beta-lactamases, or ESBL, which are produced by E. coli and Klebsiella, which leads to resistance of many antibiotics, including penicillin and cephalosporins, but also multi-drug resistant Pseudomonas. And finally, in order to reduce antibiotic resistance, antibiotic stewardship is essential. The key foundations are the following. Engage in the process. A system-based approach is essential to prevent common disease. Careful antibiotic selection, and evaluate antimicrobial drugs use practices.